Once again, class 12th bio, the same lesson, reproductive health. Today in this class, we are going to discuss about one technique that is called amniocentesis. It is very much a wider used technique nowadays. What is this technique is actually we have to know about that. It is actually prenatal diagnostic technique. Prenatal means we are able to diagnose the sexual arrangement or disorders or abnormalities, any defect, any type of the abnormalities present in the unborn baby. So we are able to define it. Amniocentesis is a prenatal diagnostic technique to detect the defect or abnormalities in unborn baby. In this topic, we are going to discuss about the abnormalities which is generated. But must remember, this is highly risky process. Highly risky process, it means it is a dangerous process. So it is important that it is performed by a specialist doctor and under supervision or medical observation. It is very much risky. Why it is risky? The question is why it is risky? In this methodology or procedure, we must note it. What happened? For this process, amniotic fluid is needed. What is amniotic fluid? It is the fluid which is present in uterus when the embryo start getting development. Various cells are there which form liquid in the uterus and that is called amniotic fluid. So what all have to do in this process? With the help of a sharp needle, doctors taken out a small amount of amniotic fluid from the uterus. A small amount. It means it is very, very small. Less than 15 ml. 15 ml or less than 50 ml. That is very, very less amount. That is taken out from the uterus, mother's uterus. It is dangerous because the needle, if it touches the growing embryo, it makes a harmful jerk to the growing embryo, and that result a great disaster in future. If the amniotic fluid is taken more in amount, then also it creates the problem for growing embryo. So it is very much important that amniotic fluid is taken in very small amount, very, very small amount first, and second, the needle does not or must not touch or jerk the growing embryo. In amniotic fluid, foetal cells are present. These cells, the cells which divide mitotically to increase the number of the cell. And amniotic fluid contains the foetal cell, which is before taken out the amniotic fluid, doctors take the sonography to see the position of the embryo and the place from where they have to take the amniotic fluid. That amniotic fluid contains the foetal cell. Foetal cell is mitotically divided, and actually when the doctor taken out the amniotic fluid, the complete chromosomal development or arrangement is completed in the fetal body. So that fetal cells are cultured in vitro. Vitro means providing the condition same as in the body but outside the body. So that foetal cells are cultured in vitro, and when it is cultured in vitro, actually it is arrested in metaphase stage. When it is cultured in vitro, it shows the chromosomal arrangement. A graph is taken out with the help of that arrangement, and that graph is called karyotyping. Here see, one karyotyping is given there. In human body, total 23 pairs of the chromosomes are present. 23 from mother's side and 23 from father's side. So this is the arrangement of 23 pairs of the chromosome. This arrangement, this graphical arrangement is called the karyotyping. As you know, in metaphase, the arrangement of chromosomes is clearly seen. Then what is next step? Next step, doctors use this karyotyping to diagnose the disorders. Disorders. Some disorders are called the chromosomal disorder, or it is also called congenital defect. Congenital, this is the one word only. We must replace one space bar from here. That is congenital defect. Chromosomal disorder, and it is also called the congenital defect. Various chromosomal disorders, the various disorders are divided into two parts. One is called autosomal disorder and one is called the sexual disorder. Autosomal, as you know, in our body, 22 pairs of the chromosomes are actually autosome. 22, first 22 chromosomes are the autosome, and the last pair is the sexual chromosome. So, if any disorder is present in first 22 chromosomes, first 22 pair of the chromosome, then it is called the autosomal disorder. One of very common disorder, autosomal disorder, is called the Down syndrome. What happened in the Down syndrome? The 21st pair of the chromosome instead of disomy, they become trisomy. Disomy is the pair of the two chromosomes. As you know, each chromosome is present in the pair. So, in 21st pair, only two chromosomes are required. But if any disorder is there, that chromosome turns to 3 in number. 3 in number, so XXX or XYY, any arrangement. If 3 chromosome is present, then it is called the trisomy. And if it is in the autosomal chromosome, especially the 21st pair of the chromosome, then it is called the Down syndrome. Sexual disorder is also detected with the help of this amniocentesis technique. Sexual disorder, one is the trisomy in the sex chromosome, that is called the Klinefelter syndrome. What happened? The 23rd pair of the chromosome, that is actually the sexual chromosome, consists of one X from the mother's side, one Y for the father's side, or one X from mother's side, one X from father's side. Insist of that. If the arrangement is XXY, then it is called the trisomy arrangement, and the disease or the syndrome that is generated due to this is called the Klinefelter syndrome. One more very much common disorder, sexual disorder, is the Turner syndrome. In this, the monosomy is occur in the sex chromosome. Monosomy, mono means one. Insist of the pair, only one chromosome is present. This is female. Insist of the XX. It is X and 0. No chromosome is here for the pairing. Only single chromosome is present. We are talking about the 23rd pair of the chromosome. So this is called the Turner syndrome. So there are two types of the disorder, that is called the chromosomal disorder, and that we are able to identify with the help of amniocentesis technique. One more important use of the amniocentesis technique is the metabolic disorder. Some metabolic disorders also generated in the body, such as the formation of various type of enzyme, excess formation, less formation. One example is phenylketonuria. It is enzymatic disorder, and so it is called the metabolic disorder. So these are the amniocentesis technique. What happens if such type of problem arises in the in the fetus inside the mother uterus? Then doctors advise for legal. We are not talking about the illegal. The doctor advice for the legal termination of pregnancy, because the coming child is not normal. So it is very much problematic to survive. So doctors advise for legal abortion. In simple word, we call it the abortion. In medical term, we call it the medical terminators of pregnancy. So doctor advice, and parents or the guardian may take the advice, and they must copy it. This is the one better side of the amniocentesis technique. As I told you, nowadays it is very much used. But why it is used too much? One is the bitter side of this technique also. What is that bitter side? That is the sex determination. Sex determination, how it is possible to determine the sex? As I told you, with the help of this technique, we are able to get the graph or the karyotypic arrangement of this, of growing fetus. Karyotypic arrangement, it means the arrangement of 23rd pair of chromosome also. What is 23rd pair of chromosome? That also we must get that information about the 23rd pair of the chromosome. 23rd pair of the chromosome, we must get the information about that also. See here, this is the 23rd pair of chromosome, and this 23rd pair of chromosome, both the chromosome is not similar in structure. Not similar in structure because by visualizing this type of the karyotyping structure, anyone or my doctor must know that this arrangement proves the birth of the male child or female child. So when a doctor take out this karyotyping, they must remove or blur this portion, that no one should able to read what is the sex of the child. Why? This is actually misuse the amniocentesis technique, which is restricted or prohibited in our country. With the help of this technique, people are able to know the sex of the unborn child, and female foeticide occur too much. And we know that if the ratio of male and female is not proper, it creates a great problem in the society. So that is actually the misuse of the amniocentesis technique. Now next is what are the reasons for increased population? The various reasons are there. You are able to illustrate all that. First is the better health service. As compared to older time, now the health services are better. So the life span is increased because of that better health service. Second, better living condition. Also people are educated, they must know, they are aware for their health, and they get the better food, better living conditions also. Third, decline in death rate. Two type of death rates are here, MMR and IMR. What is MMR? Maternal mortality rate. Mother's death during the delivery, the dead ratio of the mother reduces, so population increases. Second, IMR, that is infant mortality rate. The infant newly born baby or unborn baby, they are also say because of the advanced medical technique. So decline death rate is also one reason for the increasing population. Next, increased reproductive age of the people. People are not aware for their reproductive health. So they must take the time. At proper age, they are going for this process, that is for the reproductive process. So the child who are born are healthier, and they also increase of the population. Last is the decreased population growth. Okay, the population growth is decreases because people aware, but the number of people or population is increases. Why population is increases? Because the death rate is less. If birth rate is less, then death rate is also less. The reproductive child health programs is enforced in our country, and due to which the number of birth reduces, but the death rate is also reduces, so the population is increased. So these are the various reasons for increasing population. Now next, what are the methods to overcome this population problem? Certain methods are there, such as motivation for a smaller family. How the family becomes smaller? It is with the help of various contraceptive methods. What are the contraceptive methods? We have to learn in another class. Then advertisement in media. We must provide information from various media sources to the people about the population problem, and so they have to make the smaller family. One program is going on in the India, that is called the Hum Do Hamare Do, we two us two. That is only with two parents, only two childs are there. It means when the parents are not present on the earth, the two are replaced the parents only. That is Hum Do Hamare Do. Last is the saturated age of marriageable age. The marriageable age is increases. Increases means it is not increased by the government. According to the government, 18 is the age for the girl's age, and 21 age is for the male for marriage. But people are aware, people are educated, so they must increase the marriageable age. With the help of that, we are able to overcome the population problem. Next is, what are the characteristics of ideal contraceptive? as I told you, it is the best method for the population control. What are the contraceptive and what are the various contraceptive which is present and how it is utilized for the for controlling the population problem, that is another question. But how this contraceptive are ideal for the user? First, it is a user friendly, that is important one. Second, it is easily available. Availability of anything must provide give the approach to use it. Effective, that is very much important. What is the purpose of making the contraceptive? It is that it must be fulfilled that, so it is effective. Reversible with no or least side effect, important. It does not create any side effect on the user's body or least side effect, and it reverses its effect whenever it is not needed. And last, it should not interfere in sexual act of the user. The user who are using the act, it is not interfere in their sexual force. So these are the characteristic of ideal contraceptive.